The last technique I'll discuss is transcranial magnetic stimulation, or TMS. And this is a relatively newer technique. And this technique differs from the other techniques because here we're not recording from the brain, but we're actually stimulating the brain. So here we can actually manipulate what is happening in the brain. And then what we try to do is figure out how that actually impacts people's ability to complete certain tasks. So what happens in TMS is the following. You basically see that there's this coil here, coil that can provide magnetic stimulation to one very specific area in the brain. And where that area is, of course, depends on where you put this coil. What it does is that it then sends a brief magnetic pulse at a very high precision. It's about 1 square centimeter, so much less than a square inch, and temporarily zaps that area so that it's actually temporarily incapacitated. It's almost like you got a temporary lesion in that area. And then what that then can be used for is to ask the question, if area such and so is involved in speech, then it should be that if somebody is talking and you zap that area, now their speech is going to be disrupted. Or, if this area is involved in vision, then it should be that if we zap this area, the person reports having a distorted vision. And by the same token, if an area does not actually cause any interruption while it's being zapped, if the behavior is still the same, then it must be that that brain area is not really involved in that kind of behavior. So what's nice about this is that this is actually a way to causatively manipulate the brain and then actually study the behavior that's being interrupted or not. Whereas the other techniques are all correlational in nature. You ask someone to do a certain task, then you record from their brain and you try to correlate the particular task with the particular brain activity. So, downsides of TMS, of course, there are several. Well, first of all, it's a fairly new technique still. It's not entirely clear still how TMS changes the brain in the long run. So it might be that even though a minute after people are being stimulated in a particular area, the stimulation is gone and they seem for all intents and purposes, they seem perfectly normal. We don't know about long-term effects. Second, just like EEG, because you're applying this from the outside of the head, it has to go through the skull and it can't really go very deep down into the brain. So you can't really study, subcortical areas like the amygdala and so forth. Stimulation couldn't reach that far down, so therefore you have to stick to sort of higher level areas that are part of the cerebral cortex. Finally, for some people, this can actually cause some painful muscle spasms and very infrequently seizures. This being said, now we've gotten to a point too where sometimes people actually get a device that does something similar to transcranial magnetic stimulation and they get it implanted in their brain, and that actually helps with stopping seizures. So we still don't fully understand how this works in terms of TMS in experiments, but it's not the case that just because it's stimulating your brain, therefore it must be bad. In most cases, people walk away exactly the way they came into an experiment, and we can actually use it to study the brain. I'm going to post a video of that too, so you can see how exactly this works. So if you want to think about all of these techniques, functional MRI, EEG, TMS, what's nicest is that if you combine different techniques, you can get both high spatial and temporal resolution. You can get very precise recordings both in terms of time, when something is happening, as well as in terms of space, where something is happening. TMS is nice because it actually permits for causal inferences. We're trying to manipulate the brain and then study behavior. And all of this helps us study what parts of the brain are involved in what kind of different kinds of functions. And knowing more about that can help with rehabilitation, with teaching, with learning, and any of those kinds of things. Limitations are that other than TMS, other brain recording techniques really only provide correlational evidence. So you give somebody a task, you happen to see changes in brain activity, and then we just assume that that's related to each other, but we don't necessarily know. Depending on what your theory is that you're interested in, neuroscience doesn't always actually answer questions that psychologists particularly asks, and we'll come across examples of that in this course. And then, of course, there's limitations to the kinds of tasks and the kinds of responses that you can ask people to engage in. So in terms of ecological validity, how much are these recordings like you're doing things in real life? Well, depending on the task, more or less yes or no, right? So in MRI, if you're in a scanner, not so much. EEG, people can actually move quite a bit, so it just depends. So that's some background about neuroscientific recording techniques. I wanted to discuss that because as we go through the course and findings from neuroscience come up, it's helpful to have some idea of how these recordings, how these data points actually come about and what the logic about them is.